In the name of Allah, the Beneficent, the Merciful. Polymorphism means the condition of occurring in different forms. In genetics, a polymorphism is a place in DNA sequence where there is a variation, and that variation is present in at least 1 in 100 people. The change may be in form of one base change, or it may be in form of stretches of DNA. About 90% of human genome variation comes in the form of single-nucleotide polymorphisms, SNPs, pronounced as snips. That is, variation that involve just one base. Polymorphisms can also occur where variable numbers of tandem repeats of DNA sequences occur. These are abbreviated as VNTRs. This table shows repeats present in different loci on different chromosomes. Rflp can be used to detect human genetic variations, for example, in prospective parents or in fetal tissue. Chromosome obtained from DNA donors can be cut with restriction endonucleases to produce restriction fragments. Restriction endonucleases are bacterial enzymes. There are many different restriction endonucleases isolated from a variety of bacteria that are available commercially. In bacteria, they act as a part of restriction or modification system that protects these bacteria from infection by DNA viruses. Restriction endonucleases recognize double-stranded DNA sequences called palindromes or inverted repeats, which are usually 4 to 8 base pairs in length. This figure shows the recognition site for ECHO R1, which is a restriction endonuclease isolated from E. coli. Some endonucleases produce offset cuts within palindromes, yielding sticky ends on fragments. Sticky ends can facilitate recombination of a restriction fragment with vector DNA and are useful for DNA cloning. Occasionally, a mutation occurs in restriction enzymes cleavage site that is either within a gene or tightly linked to a gene. As a result of this mutation, the specific restriction enzyme for this site will not be able to cleave it, though it will be able to cleave normal DNA restriction site which is not mutated. Thus, two smaller fragments will be obtained from normal DNA, whereas mutant DNA will present as one large fragment. Sometimes a mutation creates a restriction site that is not present in or near the normal gene. In this case, two smaller restriction fragments will be obtained from the mutant and only one fragment from the normal. Normal human DNA has many regions that contain a highly variable number of tandem repeats, VNTRs. The number of repeats differs from one individual to another and from one allele to another. Restriction enzymes that cleave on the left and right flanks of a VNTR produce DNA fragments of variable length. The length depends on the number of repeats of that the DNA contains. The fragments produced by various restriction enzymes from a number of different loci can be used to identify individuals with the accuracy of a fingerprint. Therefore, a technique called DNA fingerprinting is used to determine parentage or other genetic relationship to implicate suspects in criminal cases. Sickle cell anemia is an example of a genetic disease caused by a point mutation. The sequence altered by the mutation abolishes the restriction site of the restriction endonuclease MST2 that recognizes the nucleotide sequence CCTNAGG, where N is any nucleotide. There is an A to T mutation within a codon of the beta globin gene that eliminates a cleavage site for the enzyme. The mutation that gives rise to this disease is actually one and the same as the mutation that gives rise to the polymorphism. This mutation results in loss of an MST2 site in beta globin gene. Samples of DNA from individuals are treated with restriction endonucleases. Normal DNA digested with MST2 yields a 1150 base pair fragment, whereas a 1350 base fragment is generated from the beta gene as a result of the loss of one MST2 cleavage site. These fragments are subjected to electrophoresis on gels. With Southern blot technique, the restriction fragments on the gel are hybridized with a radioactive complementary DNA probe for the beta globin gene. The sickle cell allele produces a fragment of 1350 bases when treated with MST2. Our normal allele produces a fragment of 1150 bases plus a fragment of 200 bases that is not visible on the gel. For a person with sickle cell disease, both alleles produce 1350 base fragments. In a normal person, both alleles produce 1150 base restriction fragments. For a carrier, both 1350 and 1150 restriction fragments are observed. Rflp analysis can be used to determine identity of individuals, though it has been supplanted by better techniques like nucleic acid testing. These are restriction fragments produced from a gene with a variable number of tandem repeats or VNTRs. DNA from 3 individuals, each with 2 alleles for this gene and a different number of repeats in each allele, was cleaved with restriction enzyme EchoR1. The cleaved fragments subjected to electrophoresis and treated with probe for the gene. The length of the fragments depends on the number of repeats they contain. An individual who is heterozygous for a polymorphism has a sequence variation in the DNA of one chromosome and not in the DNA of the companion chromosome. In such individuals, each chromosome can be traced from parent to offspring by determining the presence or absence of the polymorphism. A restriction fragment length polymorphism or Rflp has been found to be associated with a mutant phenylketonuria gene. In the case presented here, both parents are heterozygous carriers for the gene for phenylalanine hydroxylase and autosomal recessive disorder. They both have a fragment A and a fragment B, generated by a restriction endonuclease and detected by a probe. Unaffected child shows only fragment A. When DNA is digested by endonuclease, thus normal gene is associated with the polymorphism giving fragment A. An affected child lacks phenylalanine hydroxylase activity, shows only fragment B. When DNA is digested with same restriction endonuclease, therefore defective gene is associated with the polymorphism giving fragment B. When DNA is digested by restriction endonucleases, this means the fetus is affected because it has inherited 2 abnormal genes from its parents and shows genotype BB. Diagnosis for the fetus is phenylketonuria. So that is the fetal fragment. The restriction endonucleases recognize palindromes in double-stranded DNA, and AAGCTT is the only palindrome among the choices. Because sequence of only one DNA fragment is given, the base sequence of complementary strand must be determined. To be a palindrome, both strands must have same sequence when read in the 5' to 3' direction. Thus, the complement of 5' AAGCTT is also 5 to 3', which makes AAGCTT. So carry home point is that restriction endonucleases attack palindrome sequences. A base change in a palindrome can abolish or create a new restriction site, leading to fragments of different lengths.